The first part of this experiment is to extract the phycobiliproteins from the spirulina. To get started, label your glassware to prevent the confusion of its contents. You will be using 2 large test tubes, one labeled spirulina, 9 medium test tubes labeled 0 through 8, and 6 small test tubes. About 2 grams of spirulina provides enough protein for this experiment. Record the actual weight. Always tear the scale and use a weigh boat. We will grind the spirulina powder with abrasive silica powder to help free the proteins from the cells. You will need about 0.8 grams, but record the actual weight of silica. We will grind the spirulina and silica powder with mortar and pestle to break up the cells. The more cells that are broken, the more protein we will have, so grind very thoroughly. Put a lot of pressure on the pestle and grind very hard. Take turns with your lab partner grinding the powder. Be very thorough to obtain a high yield of protein. When finished, set the powder aside in a clean weigh boat under a watch glass to prevent losing powder or contamination. Now we can extract the proteins from the powder using a solvent and centrifugation. The solvent we will be using is a buffer to keep the proteins at a constant acidity. Label a beaker buffer and obtain a little over 40 milliliters. Dissolve the powder in 20 milliliters of the buffer in a large test tube. Cover the test tube with parafilm and shake to dissolve the proteins. The other parts of the cell will not dissolve in the buffer. Make sure all of the powder is suspended or dissolved. Record the color of the solution. Let the solution sit for at least 2 minutes before moving on to the next step. A lot of the unneeded cell parts will fall to the bottom. We will be using the 6 small test tubes for centrifuging. Add equal amounts of solution to each, but do not fill completely. Make sure all of the test tubes are filled evenly. Uneven test tubes will unbalance the centrifuge and cause disasters. Place test tubes opposite each other in the centrifuge to balance the weight. Centrifuge for at least 10 minutes to push the unwanted cell parts to the bottom. This will leave the proteins alone in the solution. During this time, clean the spirulina test tube. The spirulina residue may dry and become difficult to remove later. Discard the remaining solution into a proper waste container. After 10 minutes, turn off the centrifuge and let it stop spinning on its own. Do not touch the centrifuge while it is spinning. Decant the protein extract from the small test tubes into the large test tube labeled protein extract. Leave the solid behind.